Decomposition, as defined by dictionary.com, is the process of breaking down organic material, such as dead plants or animal tissue, into smaller molecules that are available for use by the organisms of an ecosystem. Decomposition is an important process in ecosystems as it determines the amount of nutrients available for consumption by higher trophic levels. According to the peer-reviewed journal article, 5 Biotic Functions of Rhiparia, leaves contribute to approximately 75% of decomposable litter on the forest floor. There are many different organisms that are responsible for the breakdown of this litter, including fungi, bacteria, and soil-dwelling animals. The purpose of this lab was to collect leaf specimens from 5 different tree species, similar to species used in the famous decomposition study titled Nitrogen and Lignin Control of Hardwood Leaf Litter Decomposition Dynamics. We would then calculate rate of decomposition, use statistical analysis to compare rate of decomposition between specimens, and compare rate of decomposition values to the literature values as found in the Melillo et al. Paper. As the rate at which litter decomposes is determined by its chemical composition, specifically nitrogen and lignin content, it was hypothesized that the decomposition rates would vary between specimens. According to the Melillo et al. Paper, white ash, Fraxinus americana, had the highest rate of decomposition, while beech, Fagus grandifolia, had the slowest rate of decomposition. As both of these specimens are similar or identical to the specimens used in this lab, it was hypothesized that when compared to the rate of decomposition for other tree specimens, white ash would have one of the highest decomposition rates, while the European beech would have the lowest. First, leaf samples were collected from 5 tree species around campus: silver maple, red dogwood, European beech, white ash, and northern red oak. Then, 5 grams of each specimen were placed in litter bags, which were constructed similarly to the picture shown in the top right. The weight collected from these specimens was recorded as fresh weight LB for litter bag. Once all the specimens were weighed and placed in litter bags, they were taken to the Olin greenhouse, where they were covered with at least 1 centimeter of soil. In the greenhouse, the specimens were kept in a controlled environment where they received mist for 15 minutes every other day, and were kept at 24 degrees C during the day and 18 degrees C at night. Then, 5 grams of leaves from each tree species were placed in paper bags. The weight collected for these specimens was recorded as fresh weight DO for drying oven. They were then placed in the drying oven, where they would stay for a week. After 1 week, the samples were removed from the drying oven and weighed. The weight for these specimens was recorded as dry weight DO for drying oven. After 4 weeks, the litter bag samples were removed from the Olin greenhouse. Any soil on the samples was removed using brushes and water. This was an important step to ensure the most accurate weight as possible once the specimens were dry. The 5 paper bags containing specimens from each of the 5 tree species were then placed in the drying oven. 1 week later, the decomposed samples were removed from the drying oven and weighed. The weight for these specimens was recorded as dry weight LB for litter bag. It was important to record all the weights of the fresh and dry specimens in order to calculate rate of decomposition. Using all of the weights collected as explained in the last 2 slides, rate of decomposition was calculated using the following equations. First, the ratio of dry weight to fresh weight for the drying oven specimens was calculated by dividing the dry weight by the fresh weight. Then, to get an estimate for the initial dry weight of each litter bag sample, the ratio of dry weight to fresh weight calculated in the step above was multiplied by the fresh weight of each litter bag sample. Then, K, or decomposition rate, was found by taking the negative natural log of the final litter bag dry weight over the initial litter bag dry weight, and then dividing all of that by T, which in this case was 4 weeks. Then, to determine if there was a statistically significant relationship between the lowest average decomposition rate and the highest average decomposition rate, a t-test was used. A t-test is used to measure statistically significant differences between the means of 2 groups and is a method of hypothesis testing. A t-test is used to calculate the p-value. If the p-value is less than 0.05, the null hypothesis is rejected and the relationship is significant. If the p-value is greater than 0.05, the null hypothesis is accepted and the relationship is non-significant. Statistical significance suggests a pattern in the means that cannot be explained by chance alone. The figure below represents the overall averages of the mean decomposition rates from all lab sections. The results show that Fagus sylvitica, or European beech, had the slowest rate of decomposition with a K-value of 0.108 and a standard error of 0.0127, while Cornus stolonifera, or red dogwood, had the fastest rate of decomposition with a K-value of 0.206 and a standard error of 0.0305. The table below represents the K-values, or the decomposition rates, as found in the Melillo et al. 1982 paper. In their study, beech had the slowest rate of decomposition of 0.008, while white ash had the highest rate of decomposition with a K-value of negative 0.47. The table below represents the t-test results that were used to determine if there were any statistically significant relationships for the highest and lowest average decomposition rates for each species in each lab group. A statistically significant relationship was found between Cornus stolonifera, or red dogwood, and Quercus rubra, or red oak, in lab section 3, with a p-value of 0.000173, and between Acer saccharum, or silver maple, and Fagus sylvitica, or European beech, from lab section 11 with a p-value of 0.00468. As seen in Figure 1, each of the standard error values for the average decomposition rates for the 5 tree specimens are smaller than 0.05. This suggests low error in the calculated mean, and also implies that the data collected and rate of decomposition from all sections had little variability. The largest standard error value was for Cornus stolonifera, or red dogwood. Red dogwood also had the highest rate of decomposition. Therefore, the error could have been caused by the difficulty in dirt and soil removal from these specimens because they were highly decomposed. If the dirt was not properly removed, this could have led to error in the final dry weight measurement, which would have impacted the calculated K-value. The following figures present the average decomposition rates between the two groups of specimens that were determined to have a statistically significant relationship between their average decomposition rates, determined using the t-test. From the t-test, we could conclude that Cornus stolonifera, or red dogwood, decomposes at a significantly faster rate than Quercus rubra, northern red oak, and Acer saccharum, or silver maple, decomposes at a significantly faster rate than Fagus sylvitica, European beech. As discussed in Melillo's peer-reviewed journal article titled Nitrogen and Lignin Control of Hardwood Leaf Litter Decomposition Dynamics, the amount of lignin present in leaf specimens is a statistically significant determinant in decomposition rates, and the more lignin present correlates with a slower decomposition rate. Based on the findings from the t-test in Table 3, lignin content was expected to be lower for Cornus stolonifera than Quercus rubra, and lignin content was expected to be lower for Acer saccharum than Fagus sylvitica. According to the literature written by Melillo, the lignin content for silver maple, Acer saccharum, was 10.1, and the lignin content for beech, Fagus grandifera, was 24.1. This is consistent with the findings in Figure 1, as European beech was slow to decompose with a K-value of 0.108, which suggests a higher lignin content, and silver maple was faster to decompose with a K-value of 0.1415, which suggests a lower lignin content. Based on our study, red dogwood, Cornus stolonifera, was found to have the overall highest rate of decomposition. In a study published in the peer-reviewed journal article titled Effects of Nutrient Enrichment and Leaf Quality on the Breakdown of Leaves in the Hard Water Stream by Royer et al., they compared decomposition rates between red dogwood, aspen, and birch. Their findings concluded that red dogwood was found to have the lowest lignin concentration of the 3 species, which is consistent with the high decomposition rates, which is also consistent with our findings from Figure 1. In conclusion, the data both supported and refuted the hypothesis. It was originally hypothesized that European beech would have the slowest rate of decomposition, while white ash would have the fastest rate of decomposition, based off of the findings published in the peer-reviewed journal article titled Nitrogen and Lignin Control of Hardwood Leaf Litter Decomposition Dynamics. As seen in Figure 1, European beech had the slowest rate of decomposition, which is consistent with the literature findings in the peer-reviewed journal article. However, white ash was found to have the second highest rate of decomposition, which is consistent with the literature as it shows that it has a low lignin content but does not support the hypothesis. According to Figure 1, Cornus stolonifera, a species not studied in the Mulilo paper, was found to have the highest rate of decomposition. Other studies found that Cornus stolonifera had a low lignin content, which supports the findings that suggest a high rate of decomposition. If the lab were to be repeated, it would be pertinent to ensure that the soil and dirt were thoroughly removed from specimens that were more decomposed, as the standard error for the mean was higher for these samples.